First of all, it's quite an interesting molecule and a quite recently developed molecule in the field of hematology, because our group was one of the groups which reported the importance of this particular antibody in acute myeloid leukemia. So it is being highly overexpressed in AML, approximately 40% of patients, and researchers focused on CD200 has shown that it is also being expressed on the surface of leukemic stem cells, which are key important targets for leukemic therapies and are responsible for the relapse. So my study basically is focused on the evaluation of a monoclonal antibody that targets this particular CD200 molecule, and it has shown very good results in preclinical models on cell lines and patient samples, as well as mice models, that antibody actually blocks the interaction of CD200 with its receptor, that is pretty much a potential therapeutic target in AML. So in the term of patients, like this antibody, there are different kind of monoclonal antibodies which has been developed for other cancers, especially solid tumors, but hematology oncology lags behind in the development of the monoclonal antibodies regarding, and it's their clinical implication and FDA approvals in terms of first-line therapy or therapy in the consolidation or other phases of the treatment to patients. So in this scenario, so if the patients are expressing high CD200 on their AML cells, and we treat those patients specifically with this particular antibody, so it might help the patient for long-term remission. That's what the ultimate aim of any immune therapy is. So it's targets a broad number of patients than specific mutants of specific proteins. So this targets broad number of patients, and then it also helps not only in the first-line therapy, it may also help in long-term remission of the patients who have refractory disease or may have a chance for high relapse rate in AML. Definitely, we do have plans for testing this agent at least in phase 1 clinical trial, because we have done a lot of preclinical experiments using the cell lines, using the AML patient samples, as well as the in vivo models. So this whole work was a part of MRC funded project, and the next step of this project was to go into the clinical trial committee for the approval of first phase clinical trial of this particular antibody from Trillium Therapeutics that we can use on the patients, especially which have high expression of CD200.